So just to give you a brief presentation of Professor Cremer, he's, as the director said, is at the University of Munich in Germany, and Professor Cremer is one of the most known scientists working on the organization of chromosomes inside the nuclei of the cells, and he has developed, I can say in collaboration with his brother Christoph, new tools to understand how chromosomes work in cells and in different conditions, and he's very known for having developed a picture of the chromosome as being organized like countries on a geographical map. So I'm sure that he will show very, very nice pictures about that. And he has done many other works on gene expression and gene regulation during the course of the cell cycle. So we are very looking forward to hear about your talk.
Yeah, good afternoon, everybody. Everybody can hear me also in the back? Yeah, good. Yeah, it is a great pleasure to be here, and in particular, we spent a morning with wonderful lectures and discussions. I had a little workshop on it, and this is, I think, the first time I came to a place and not just giving my talk, but I had really a very close interdisciplinary communication this morning, and this is necessary when you want to study the nucleus. Now, the nucleus is a telling example of a biological system where changes of structure may precede or follow changes of function. Understanding the functional nuclear organization in space and time depends on understanding its structural functional interactions at all levels of organization, from molecules to the entire system. Now, what is the long-term goal? You know, we have now the genomes. People are doing genome sequencing, decoding the DNA sequence across species and cell types, not only the coding regions, but also regulatory sequences and other regions which may be just involved in organization and higher order architecture. There is the epigenome with its goal of decoding the chromatin language in space and time across species and cell types. And when you have one diploid genome in your body, you have many epigenomes in all these cell types in differentiating cells. And there is what I call the 4D or space-time nuclear architecture, a higher order chromatin organization relation to the dynamic topography of nuclear bodies, such as splicing speckles, machineries for transcription, co-transcriptional splicing, DNA replication repair, down to the dynamic positioning of single macromolecules, DNAs, RNAs, proteins. And the goal is the decipherment of universally valid as well as species and cell type specific rules of nuclear architecture and their evolutionary origins. And we think that the term nucleome in space and time may integrate these levels with the goal of understanding evolutionary, structural, and functional integration of genomes, epigenomes, and nuclear architecture in space and time, and to understand how nuclei function. And understanding the nucleome requires an interdisciplinary approach. Clearly, you need epigenetics, genetics, you need nuclear biochemistry and biophysics. You need to understand nuclear environment, starting from the cytoplasm around the nucleus, cell membrane, other cells, tissues, organisms, and the impact on the environment at large, which may affect the epigenome and may have consequences on the nucleome in space and time. And I think what is least known still is the overall nuclear architecture, its evolution from prokaryotes to eukaryotes. Now, in the beginning of the 20th century, even the late 19th century, people like Theodor Boveri, like Karl Rabel, like Eduard Strasburger, thought that chromosomes in a nucleus may be organized in what Theodor Boveri called territories. And these territories may be built up from some clustered chromatin. And you see, time goes on, and in the 90s, textbook pictures told students, okay, you understand the DNA, how it's wrapped around nucleosomes, 30 nanometer fiber. Forget about the rest, that's just a kind of spaghetti, and we do not need to know how this is all arranged. Now, it was already mentioned that we developed methodology to visualize individual chromosomes here in a fibroblast, human fiber nucleus in different colors, and we started to be able to measure distances, how homolog chromosomes are arranged, and so forth. And it is based on a color scheme where probes, so-called paint probes for each chromosomes, were marked with different kinds of fluorophores, and you see that a number of chromosomes can carry, the probe can carry one fluorophore, that we can nicely distinguish the chromosome territories in such a multicolor approach argues there cannot be too much heavy intermingling of fibers all around, otherwise this method would not have worked. Now, nuclear architecture. Generally agreed is now, yes, that chromosome territories exist, that there is a 10 nanometer fiber since the 70s, the Ollins works showed it with the nucleosomes, but there is a big gap of firm knowledge in between, and there are highly controversial discussions about how the 10 nanometer fiber is organized into chromosome territories. Now, we know a lot about the distribution of genes along individual chromosomes. It's not uniform. There are clusters where there is gene-dense areas and others where gene-poor areas. And when you take mitotic chromosomes and hybridize ALU sequences to it, here it's a DNA counterstain, you see that ALU is very non-uniformly distributed, and the ALU-rich regions, they correspond to gene-dense regions. And we have then taken Buck probes for those regions, for gene Bucks with segments for gene-poor regions, and Bucks with segments for gene. Sorry, this should mean gene-dense regions here. This gene-poor region, green, is gene-dense, and this is a reconstruction of a lymphocyte nucleus where you have two chromosome 12 territories, here as neighbors, and you see that the gene-dense material is exposed to the interior of the nucleus and the gene-poor material in the periphery. And this is quite a robust finding, this non-random radial organization, and has been found in many examples, many cell types, and also in species from mouse, men, to, say, Hydra. Now, another method to visualize. I would like, please, could we bring down the light? Please switch it down. You see it much better. You can use thymidine analogues for incorporation into the DNA, which are directly labeled with a fluorophore. And when you pulse, this is even worse. Okay. It would be helpful if you could take away more light. I mean, in spite of the afternoon, it's a little bit dangerous, but I think we can take the risk. Because I have movies and so forth. If you take, say, a cyanine 3D UTP and make a pulse labeling in early S-phase, you get many of these little replication foci with a size of roughly 1 megabase. And simultaneously, you can express a histone here 2B, which is tagged to a green fluorescent protein. So you can see in the live cell the chromatin and these replication foci. Now, when you do it here in S-phase, you label daughter strands in both in both chromatids, and you have mitosis where the chromosome is labeled with both chromatids. But when you now go to the next cell cycle, to semi-conservative DNA replication, you see that in the next mitosis only one chromatid bears the fluorescent labeling, the other is non-labeled, and this is then distributed randomly, labeled and unlabeled chromatids to the different daughter nuclei. And once labeled, these strands are retained, so they are distributed over the growing clone, and it's obviously very difficult to switch down lights. Please, I would really love to have these things off. Or are we sitting then totally in the dark? I'm not sure. It's an experiment. You see, this is a nucleus fixed 3 cycles after the labeling. And you now see individual territories which still show these replication foci, which are maintained over time. And here you can start, I will show you later how territories move, but it's interesting that once labeled, these replication foci appear not only in S-phase, but at G2, in the next cell cycle, everywhere during the life cell of these growing cells. Now, this has brought us to this kind of model called the 1 megabase chromatin domain model. And you see here how these chromatin domains, which are replication foci, act as during S-phase. Oh, that's very nice, thank you. Have locally constrained movements. When here you have a model nucleus with different colors, when you have the same color and you make virtual sections, you see there is space in between, what we call the interchromatin compartment, but you could not distinguish individual chromosome territories. Now, more recently, we have used EDU pulse labeling, and this is an early S-phase nucleus, and which was recorded with structured illumination microscopy, a new high-resolution method. And we counted some 6,000 replication foci during early S-phase, and when we take the entire interface, we may come to some 10,000 of these foci, meaning each focal domain may have something between 500 and base pairs and a megabase pair. Now, other methods have recently shown from the Mishima group, for example, and from Elsoff, that with cryo-electron microscopy and an X-ray scattering, you can argue that these chromatin domains are polymer melts, so where the nucleosomes are so close that a 30 nanometer fiber cannot be retained. And here you have nuclear pores, you have channels in between, and arguably transcription takes place at the surface of such domains, and I will show you more evidence for that later. Interestingly, a completely different way, namely called chromosome conformation capture. I'm not going into the details. It is published, first paper in 2002, and very interesting paper since 2009. Combined with deep sequencing, you can measure the contact frequencies of DNA in a nucleus or in a population, say, of 10 million nuclei, both within a chromosome and also between different chromosomes. And I just show you here only a little piece, a syntenic region of 3 megabase pairs in mouse and man. And these clusters here are contact frequencies which show you this is a domain. And what we see in microscopy has been confirmed with molecular biology, and they call it topologically associated chromatin domains. Now, this has led to more modeling, and Leonid Mirny claims that it would be very helpful to organize such a megabase-sized domain in smaller globule subchains which take certain parts of that domain. Forget about fractal, I'm not going into that, but the point is that when a contiguous stretch of DNA packs into a compact such structure, it's unknotted, and it makes it easy to pack and unpack, to open and close. When you have an organization with global subchains which are extended throughout, you get have a highly knotted shape which is prohibiting this kind of unfolding. And Gregor Kret, in my brother Christoph's group, a physicist, has made such models in 2000 already, where each dot is a nucleosome, and we were questioning, well, how could that organization be, and could it be this kind of random organization, or could you have these little globules, or how we call it, 100 chromatin domains. Now, I would like to show you at least these following two slides to show you how you can do live cell experiments. And here you see a living cell nucleus where part of the chromatin is either labeled in red or in green. And you see this goes through a mitotic event, and you get two daughter nuclei. How was it done? Here, in one S phase, the cells were pulse labeled with a thionine 3 DUTP, and in the next cell cycle, the same cell was again during S phase labeled with a thionine 5 DUTP. The total chromatin was visualized by an H2B tagged to GFP, and these, of course, are false colors from the digital recording. Now, when you do this, during further cycles, you get a segregation of green and red chromosome territories. And what you have seen here are locally constrained movements of the territories. We have not observed that the territory can move from one side of the nucleus to the other, but these local movements. Now here in the second slide, show you another method which we have developed. When you express a photoactivatable GFP tagged to the histone H4, then you can activate that GFP by laser microirradiation with 440 nanometer. And the total chromatin is here shown in red by expression of H2B tactune MRFP. Now with the microbeam we can do lines and visualize that. We can make this kind of markers over the nucleus and see what happens with time. This is an embryonic stem cell nucleus from the mouse, and regularly see within 20 to 40 minutes there's lots of changes of nuclear shape and movements going on. When we do the same experiment with fibroblast nuclei, they are called potatoes. They are not interested in this kind of movements. And we do not know what the difference is, but just to show you, don't look only on fixed cells, look on the dynamics. Now, without explaining how the method works, I come to 3D structured illumination microscopy. When you do a conventional confocal laser scanning microscopy, this is the point spread function, and you have a resolution, say, of 250 nanometer laterally, and here 500 to 7 nanometer axially. When you use structural illumination microscopy, you reduce the focal area quite a lot, and this results in an 8-fold increased resolution in 3D compared to conventional confocal microscopy. And now I show you pictures and experiments based on 3D structural illumination microscopy. Here you see a flat nucleus, and this is the chromatin in green, and in between you see space. You see clusters of very intensely stained DNA, and when you go to the periphery, you see a kind of channels going outside. And when we do reconstructions of nuclei, we see here in green, this is what we call the interchromatin compartment, that these little channels go to the peripheral layer of heterochromatin, and here we have used an antibody against the nucleopore protein, and we see that the little channels are kept by these blue signals for the nucleopores. So the channels end indeed at nucleopores. When we use peripheral sections through a nucleus, where we can see here like black dots, these little channels, and we stain H3K4 3-methylated as a marker for transcription competent chromatin, we see that these markers often seem directly attached to the nucleopores. When here in the same experiment we use a marker H3K27 3-methylated, a marker for silent chromatin, it's a little bit removed at these places away from these pores. So the nuclear pores, we think, and the channels there, they, I will show you later, are part of the active nuclear compartment. Now, people think that DNA is just extended through the nucleus. That's the spaghetti model. But when you specifically stain the DNA for electron microscopy, then you can do serial sections and reconstruct a nucleus. And when you see on the entire nucleus taking away just the top, everything seems to be filled. But when you look on the thin sections, you see how much space is there in between. And in the periphery, we see these little holes, which are the channels going to the nuclear pores. And when you turn it around, you may see even better. Yes, there is a lot of space in between the chromatin, which is a kind of clump. Now, I want to show you two examples how we can use the methods to do biology. Both are unpublished. and it's about changes of nuclear architecture during cell differentiation and development studied with 3D super resolution microscopy, in this case structural elimination microscopy. The first example is about the bar body architecture in most somatic cell nuclei and the bar body development in female embryonic stem cells during differentiation. And the bar body, as you know, is that inactive X chromosome which every woman bears in each of her somatic cells compared to the active chromosome. The second example are changes of nuclear architecture in early bovine embryos after in vitro fertilization during major genome activation. So a little bit of introduction in placenta mammals. Gene doses compensation between XX and XY is achieved by the silencing of one X chromosome in female. The onset of X activation at the blastocyst stage has a random selection of a paternal or maternal X, and after inactivation they have clonal transmission, and that's a paper under review with the data. Now, here you see a nucleus stained with DAPI, for it's colored in red, and you see here this clump of chromatin. And when you do a painting for the X chromosome, you see this territory, which is in 3D reconstruction quite round, while this territory, X territory, is much more extended, flatter. Also, the volumes are not so much different, maybe a factor of 1.5 or 2, but not more. Now, when you take these markers for transcription competent and repressed chromatin, and here you have such a bar body, you see it's very much enriched in repressed chromatin, and competent chromatin is only mostly seen outside with a few exceptions. When you have now to come to another point, from the inactive X chromosome, you have the expression of a so-called cis RNA, a non-coding RNA, which seems to be important in the inactivation process. And it's transcribed first from both chromosomes, but then only from the X chromosome chosen for inactivation. A 70 kb long transcript with a half-time for 4 to 6 hours, and it's strictly acting in cis. And what I mean, you can see here, this is the nucleus with the conventional wide field resolution, a clump of cis RNA seen here. But when we go to structural elimination microscopy, we see that cis forms a little dots throughout this bar body. And you see what it is now. I have to go back. I hope I can get this. This is now a reconstruction of a nucleus which has 3 X chromosomes and 2, as you have seen, inactive ones. And you see how these multiple cis foci are distributed throughout the inactive X territories. When we look in more detail, we see that these cis RNA foci are associated with sites which are a little bit less intensely stained with DAPI, and we think that these, since they arranged together as a network, could form a collapsed channel system. Now I come to the question how this inactive X is established during early differentiation of female mouse embryonic stem cells. And it's known that cyst RNA is starting here when you initiate the inactivation process between day 1 and day 2. Then you have transcription silencing on one of the chromosomes, and you start to see modifications of histones, X3 as CON4, and you come to irreversible inactivation associated with the formation of macro H2A and DNR methylation events. That's a scheme from Edith Hurt. But the time course of chromatin compaction in the designated inactive X was not known, and we have studied this. Here you see a nucleus from such an embryonic stem cell at the moment. I think that should be at day 2 or so, yeah. And you see that both X chromosomes show cyst RNA associated with RNA pol 2 here, and you would not see any difference between the two X territories. When you go on around day 4 after the induction of differentiation, cyst RNA has spread in the entire area. The RNP2 clusters are now seen mostly only at the periphery, and we see the start of the compaction process. And here at day 5 we see the bar body compaction as it's seen in normal somatic cells, and you see here these multiple clusters of the cyst RNA foci. Now we have achieved this model idea. Here is a nucleus, a section SIM. Here is the bar body. This is an area beside which has, of course, chromatin from some transcription competent territories. And I have not shown all the data, but this is a general view. We believe that the transcription competent chromosome territories have an interchromatin compartment system lined by transcriptionally competent chromatin, which starts at nuclear pores and then extends into the nuclear interior. We can also find that markers for nuclear matrix proteins like SAF-A or matrin-3 are in the interior or the periphery of this interchromatin compartment system. In the bar body, that system has collapsed. There are these in blue, the cyst foci, occasionally an RNA pol 2, but only very little. There is a SAF-A which has an X-specific modification, and I have drawn this to show the limits of the structural illumination microscopy, that the sizes show you the resolution limit. And below is what may be, what we may find at higher resolution. We think there's the channel system that some chromatin in the active compartment may go with little loops into the interior. Here should be transcription going on, and here you have the collapsed system. Hopefully, and I will show you this at the end of the talk. Here we have now a resolution of about 100 nanometer lateral and 300 nanometer axial, but there's hope to come to a resolution of 30 nanometer lateral and axial with the new methodology. I switch now to my second example. I see it's known here, example 3. I put something out, but changes of nuclear architecture in early bovine fertilized embryos during major genome activation. For introduction, I want to say, look, you have during oogenesis an accumulation of maternal RNA and protein. Then you have the fertilization. Still you have the maternal RNA and protein. A lot of that maternal RNA is going to be used to make more proteins. And at the 8-cell stage, there is the start of the major genome activation. The genome is going to be activated. The maternal RNA is degraded, and now the embryo has to live with what coding information it can produce by itself. And we have focused on this window from 8 cells, say 5 to 8 cells, to this 16 cells to an early molar stage to see what happens with nuclear architecture. And it was really surprising. This here are 8 cells, 8 nuclei from an 8-cell embryo. And these are midplane sections with structural elimination microscopy. These are top sections and bottom sections. And you see that the chromatin obviously associates at the periphery, having a large lacuna in the interior where we cannot detect DAPI, stains DNA. Also with other DNA stains, we don't see things here in the interior. And then here such a 20-cell embryo, and I'll show you just examples. You see here is one nucleus which has still this pattern. But the other nuclei show now chromatin dispersed through the interior. Here's still a little bit of a lacuna, but in general, nuclei get smaller. And when we look in higher resolution on what happens, there are movements of these clusters of chromatin in the periphery to the interior. These major lacuna disappears, and we see also that now nucleoli appear which are surrounded by densely DAPI stained material. And when we look on here this quantitative evaluation, we see that in the 5 to 8 cell stage, till the onset of 90% of the nuclei show this pattern which we call ENP, embryonic nucleus, with a peripheral organization of the chromatin. During 9 to 16 cell stage, we see a predominant phenotype of nuclei which shows us that this chromatin is moving into the interior, and in embryos with more than 18 cells, we see a conventional type of nuclear organization with chromatin expanded throughout the nucleus surrounding nucleoli, what we typically see in somatic cells. During that period, the nuclear volume decreases highly significantly. And you see in the intermediate stages, they are still relatively large, arguing that the movement of chromosome territory is not just enforced because the nucleus is made smaller. They start to move first, and then the nucleus is going to decrease in size. Now, to our surprise, here you have again an embryo here with 7 cells at the onset of EGA, and here you have an embryo with also some 20 cells where you see this occasional type which has been retained here, but the others nuclei are small with this conventional organization. And this is now a cloned bovine embryo. Sorry, EOF is wrong here. It's a clone. It's obtained after by the transfer of a bovine fibroblast nucleus into an enucleated bovine oocyte. And that's interesting that a nucleus, a fibroblast nucleus, which is an elongated, rather flat nucleus, is forced in this environment to go through the same changes of nuclear architecture than the fertilized embryos. And you see here by chromosome painting, this red thing is just a gene. We look at painting shows that these clumps of chromatin are individual territories, then the territories move into the inside, and a typical organization of a nucleus is made. Now, how can that happen? The nuclear envelope gets smaller. You see, it's maybe I think not large enough, but here you will see we can feel invaginations from the nuclear envelope. Here we have stained lamin B, and we find that this can be invaginated. There can be excisions of the lamina, which are either removed outside or in the interior. And you see there is no DNR associated with it. We think this may be a glimpse what happens, but the mechanism must be much more complex. Now, another question was whether in these so-called ENPs, which are in the 5 to 8 cell stage, is it a synchronization? Here you see from the same embryo a nucleus pulse labeled with EDU for DNA replication, and you see the periphery is pulse labeled, it's in S phase, and here another nucleus stained with H3S10 phosphorylated, a marker when the cell goes into prophase. So these nuclei with this phenotype exist side by side. The next point, when one look in these so-called ENPs, nuclear peripheral organization, we see that here precursor nuclear bodies are being formed, and during the transformation process after the onset of EGA, we get large internal nucleoli, and which are just here visualized by B23, a marker which surrounds these nucleoli. Maybe even more interesting is the fact that already at the 8-cell embryo, this is another complete embryo, all the nuclei show the peripheral chromatin is enriched in H3K4 3-methylated, arguing that this is transcriptionally competent chromatin at this point. And here you see what's in the interior of such a nucleus, these large lacuna. Lots of splicing speckles are around, and we can prove by labeling with BRUTP that already at this stage nascent RNA is being formed. It's a 40-minute pulse, so this is not the site of the formation, but we think it has moved from the periphery to the interior. And now look on a nucleus after major genome activation. Here you see splicing speckles. You see the strong RNA synthesis in the nuclei, and this is what happens in this closed window. So this is what we think at this moment. There are viable embryonic nuclei with a peripheral chromosome territory distribution. When they march through major genome activation, they get smaller. they form a conventional architecture, and this is the viable population for proliferation. And some of them may still undergo apoptosis, but these, when they are not able to make this transformation, seem to be non-viable and they seem to be eliminated. And we have now also some data that they become tunnel positive and go into apoptosis. Now, here I come back to structured illumination microscopy. This is a nucleus obviously starting its transformation. It has already formed in another section here nucleoli surrounded by heterochromatin. But you see these clusters of chromatin stained with H3K9 3-methylated in red or H3K4 3-methylated in green. And when you look on the pattern of distribution of DAPI stained DNA, you see that the silent chromatin, as expected, is more at the periphery, and here is transcription competent chromatin, here also in green, and from inspection of many pictures, we had the feeling that the transcription competent chromatin is exposed to these areas which belong to the interchromatin compartment. Now you can do also a simultaneous immunostaining of H3K4 and polymerase II phosphorated at serine 5 of its C-terminal domain. That is a modification necessary to start transcription. And despite this dramatic change in size and distribution of the chromatin, what we see is that clusters of RNA pol2 shown in red and H3K4 in green are located in the periphery of these densely stained clusters. And black pixels here show you a co-localization. Typically, we see associations of transcription competent chromatin with these clusters of RNA pol2, but we see also clusters of RNA pol2 which are free. Now, what we have done is then taken nuclei and made DAPI intensity classes from 1 to 7 with the lowest intensities and the highest intensity, and compared the co-localization here of an H3K4 3-methylated chromatin, here of an RNA pol2 with these different intensity classes. And here you see the quantitative evaluation of each 20 nuclei, where we used, say, 4 to 6 central sections of nuclei with structured illumination microscopy. And we tested the following null hypothesis. If the RMP2 signals, or the H3K4 signals, or the H3K9 signals would be randomly distributed without any connections of the DAPI intensity classes, then we should see, of course, the same fraction of RMP2 positive pixels as the fraction of DAPI intensity pixels belonging to a certain class. So, if the null hypothesis would be true, we would get here a line. And what I show you here are the excesses of RNA red, RNP2, H3K4, in red H3K9, in green H3K4, and in blue RNP2 signals. And you see that in the highest intensity classes there is an excess of the marker for silent chromatin. there is an underrepresentation of RNA pol 2. You have an overrepresentation of RNA pol 2 and also of H3K4, the marker for transcription competent chromatin, competent chromatin in the periphery of the densest classes. And this is what we see both in nuclei studied at the beginning of major genome activation and in nuclei after the which have finished major genome activation. You see the same pattern showing you the distribution of the signals is highly non-random. And here I come to our present model, which argues that there are these little channels from the nuclear periphery and the nuclear pores expanding here in both types of nuclei to the interior, and H3K4 is enriched at the periphery, and the compact parts of the chromatin, the silent chromatin, is more in the interior. And we argue that what is red here would be the inactive nuclear compartment, and what is shown here green euchromatin, and also the interchromatin compartment is what we call now the active nuclear compartment. Here in the interchromatin compartment we have splicing speckles, nuclear bodies, machineries, and all these stuff can interact with the chromatin which is transcription competent in the periphery, where you find your RNP2 polymerase enriched, and where you can also show the formation of nascent RNA. I want to end my talk with a few perspectives. There is the development of spectrally assigned localization microscopy, and it means a 3D mapping of fluorophores detects individual proteins or nucleic acids at the nanometer scale. And the principle of this method, which was first published by several groups in 2006, can be explained by the French painters who did pointillistic pictures. Seurat, that bridge, when you would have looked on the painter doing it, he made little colored dots, and you see nothing. The more dots, the smaller the dots, the higher the resolution of that picture. And that can be done by microscopy too. And this is from my brother Christoph's group, a development where they use the ability of fluorophores like GFP, like Alexa, and so forth, to blink. That is, when you take the right wavelengths and the right intensity, it seems to the medical doctor that's the end of it. I mean, you have bleached now your preparation. But if you do it correctly, a few fluorophores start stochastically to blink. They emit a few hundred photons. Now you have to do this in the way that an individual fluorophore, which is just blinking, is optically isolated. That means the next fluorophore should not be closer than the upper limit of resolution. If you do this and you take a picture, then you can use these photons, hundreds of photons, to precisely pinpoint the localization of the fluorophore. Now, when a fluorophore, which is say 20 nanometer away from it, emits in the next image you take, then you can do it separately, and then you can put and record all these individual mapped localizations together, thousands and thousands, and you get a highly resolved image. And this can be done with blinking, and in collaboration with my brother's group, I show you this picture. Here you have GFP tagged H2B, and you see the size of each dot is calculated in a way to represent the precision of the localization. Few photons, broad dots, many photons, smaller dots. And you see here clusters of RNA pol 2 in these areas between the chromatin. Now, I have to say, here we take an indirect immunofluorescence microscopy or staining. That is, the fluorophore is tagged to the second antibody. That means if you do not wash your specimen perfectly, you get, of course, a lot of background. It would be nicer to have a direct labeling of your protein of interest, but it brings you to a resolution in the range of 20 to 30 nanometer, and there are tricks also to improve the axial resolution by some astigmatic lenses. So you can expect that the structural illumination microscopy with 100 to 300 nanometer can go down to a 30 nanometer resolution. And this is the most recent paper from Christoph's group, a combination of structural illumination and single molecule localization microscopy in one setup. And you see here side by side the wide field recording, structured illumination microscopy, and spectral spectral position determination microscopy with individual singles, showing you how you can improve the resolution. I would also like to point out that super resolution fluorescence microscopy in the future will allow imaging of living cells. And here is one example with structured illumination microscopy. This is a midsection and an apical section to a HeLa cell nucleus where protein, where the histone H2B was tagged to GFP. And we see the same pattern here in the live cell as we can see it after fixation. And this is very comforting to us that one can maintain this ultrastructure throughout the fixation procedure. This is the last slide. The general perspectives are there are cell population assays, in particular from molecular biology, but they are also trying to come down to single cells. There are single cell assays. Advanced microscopy is a beautiful approach for single cell assays, and you need the combination of molecular biology and advanced microscopy to better define what's going on in life. And this would be the goals of advanced microscopy in 3D space, in 4D space-time, to have a cartography of nuclear constituents at the low nanometer scale. Now, I have to acknowledge many people, in particular those in yellow, my wife, Marion Cremer, Johanna Markage is a postdoc, Barbara Hüttner and Jens Popken as doctoral students have contributed heavily to the results I showed you. Jacques Rouquette, together with Stanford Kahn, as a visiting professor, developed electron microscopy, which we are using in parallel now also as correlative microscopy, starting with live cell, going to fixation, having structural illumination microscopy, and finally electron microscopy. Sabrina Rosberger is the lead author of the new instrument I showed you. I couldn't go into the data with Sergio Ferrari, Mariano Lomiento. This is about the differentiation of hematopoietic cells. Eckhard Wolf and Valery Zakharchenko, he is really a cloning expert with when we worked on the bovine embryos. Lothar Schermele was my doctoral student. He's now in Oxford, and with Daniel Smith, PhD student, and in collaboration, in particular with Marion, they did most of the data I showed you on embryonic stem cells, and there's Volker Schmid, who helps us with statistics. Thank you for listening. Thank you.
Can you hear me?
Yes.
Okay, so first question is about the gene movement and the movement of chromosome territory. So do you think that it has been shown for some genes that can move through actin polymerization inside the cells? Do you think that chromosome territories also move using the same mechanism?
One should be very, very cautious in thinking that everything has one and the same mechanism, what's going on. But in these embryos, in these nuclei, you see still nuclei which is still quite big size, but the chromosome territories have moved into the interior. So this is for me a good example of large-scale movements of chromosome territories. Now, how it's really possible, I do not know. One possibility would be that you have some actin-myosin systems inside which pull it to the interior. We have not done stains for these compounds. There is a claim, published evidence, that even in fibroblast nuclei, when you bring them in serum starvation, that some territories may move from the peripheral to interior and some in the opposite direction. We have, you remember that experiment with this histone H4 tagged to photoactivate with GFP, and Jens Popken has illuminated the edge of fibroblast nuclei, and then we have done these experiments and thought if something is moving to the interior, it should take with in this marked fluorescent chromatin. We couldn't see any effect. So one has to be careful when studies are based just on fixed populations and evaluations. So that's one problem. Now, with respect to small-scale movements, which are constrained movements, locally constrained movements, I think, yes, maybe there is a mechanism with some actin or so doing it. It becomes much more complex when you think about data which propose that genes on different chromosome territories can come together, what is called gene kissing. The reason is, I didn't show the data, but we have done these patterns on daughter nuclei. We have followed it through the entire mitosis. We see that the pattern is changed. The daughter nuclei have a similar pattern, but this is from the anaphase movements, but that pattern does not reflect the mother nucleus pattern. So there is a lot of variability in the neighborhoods. And it's like people standing in a dark room, who do not know where the other is, How do you go? In which direction do you extend your hand, If that's true, it must be an extremely complex mechanism. You have also to think about the outside. In the embryonic stem cells, we see dramatic changes all the time of the shape. So something can if you have something and you push things together, maybe you get something which comes together which is apart. We have also seen rotational movements, and when you have a flat nucleus, and you have a movement, a rotation of that nucleus in such an axis, and we have proven it by activating parts of the chromatin, and we see that when the nucleus rotates in this way, chromatin far away from each other comes close to each other, is separated far away during these kind of movements. So there can be mechanisms from outside, from inside, just the point is we do not understand them.
And you never tried to use any of the drugs that can inhibit actin polymerization, for example?
Yeah, that people have done it, and yes, I would love to do it, yeah. I mean, but we haven't done it.
Yeah, because I have used some of the drugs, and this is why I'm asking.
If the drugs are as specific as people typically claim, so I do not know, you know.
And especially in embryonic cells, it has been shown recently by a very nice paper in Genes and Development by John Gurdon that nuclear actin is actually involved in silencing of OCT4.
Oh yeah, I mean, just I think it's important that you have solid observations in the first place, what's going on, in particular with large-scale movements. And then on such a model, you can start to try to figure out the mechanism. Of course, yeah, and it will, I mean, Andy Belmont has worked a long time on movements, and he shows that there can movements be where a clump of chromatin kind of jumps several microns to another side, and it does not seem to be a kind of random path. It seems to be directed. How should it be directed? Maybe the channel system which we see helps to guide some movements of chromatin along channels, but again, it's not known.
Okay.
One question from Professor Malamaci, and then another one. Just a curiosity, did you ever try to see if preferential clustering of different territories belonging to the same chromosome within the same locale may depend on proper expression of long non-coding RNAs stemming from this region? Thanks.
No. I mean, you know, my answer to this is the following. We are very much engaged to develop the methodology to see what's going on with the improved microscopy and so on. And what you are asking for is a person who plays the viola, could you also play, say, the tube or the trumpet? Anyway, this is a question of chamber music, with people who are really able to do the other things, and you have to come together and to do it together, that's the only way. We would not be able to develop simultaneously in a little group all the other methodology, even if it's doable in principle. I have learned to play an instrument really excellent. It takes much time, and you better don't think you can do everything alone in one group.
Thank you. Chromosome translocations between chromosomes belonging to different territories could be an interesting system to verify and validate and monitor this movement of territory. Is there any evidence that translocations somehow correlate with the movement of territories?
There is, say, pro and con evidence. People argue that chromosomes, which in the case of the development of a tumor cell type or also in evolution, have a higher chance to make a recombination or so, might be closer to each other in the nucleus. The other question is to what extent could it be just a selective fact, something gives an advantage, growth advantage in some cell type, not in the other. And it's difficult for me to be sure what's right. A problem of all these fish experiments is you base your evidence what happens on a population of cells, and there are it's not so easy to avoid bias in the evaluation, and I'm not quite sure whether. I think some of the data we have in the published literature may be a victim of a biased evaluation. It would be, in this case, it would be wonderful if life cell things, yeah. When you really, maybe the best example I know of is Edith Hurt's evidence that the two X chromosome territories during this inactivation process seem to move together, and she argues that a kissing effect is part of the inactivation process, yeah. And ideally would like to have markers on both X chromosomes and really show it in a life cell situation. Look, here's the nucleus, and 5 hours later I show you here they have come together. That would be the fantastic experiment, and I think it will come.
We have two last questions, one from Professor Gustincic and another from Professor Silvio Onesti, and then Professor Cremer will be around for the afternoon. Let me also remind you of that.
Thanks. So I was very happy that Professor Bruschi made this question, because there was a paper in which translocation was actually preceded by the expression of a transplicing transcript.
Mm-hmm.
It actually was really, you know, transcribed from the two regions that were involved.
Mm-hmm.
So I was wondering, what is your feeling? Could be that some transpliced small RNA can actually participate in the regulation of the chromosome territories distribution?
I think in the introduction, I think I tried to point out, yeah, that one should understand the nucleus as a biological system, yeah, and try to come to it with all kinds of methods. When you do so, you need to develop kind of gold standards. Yeah, you need to know, okay, this method, this way, this a good way to apply it. The people are aware of all the pitfalls, and when somebody says, "I'm not going to use the gold standard," he has to say why his way is better. Going to all the fish data, I cannot judge from the literature whether something is good or not good. Why? When I say people have evaluated 50 nuclei and there's a highly significant difference of something, I do not know how many nuclei have been evaluated, how many nuclei have been regarded. I just know when, say, a student who looks through something and he says, okay, this is a bad nucleus, too much background or so, and this and that, there is an easy chance that he ends up with a highly significant biased representation of the nuclei. In particular, we need really to understand the variability. Even if there is an average value where something is smaller or so, yeah, you need really to see the cell-to-cell variability to come to the point where you can really think about what it means. And again, I think more life cell imaging has to be developed. Look, there was a recent paper in Science from the Misteli group, and they have really shown when you have double-strand breaks, yeah, and very specific sites, and they have stained the environment, that they could follow the movements in a life cell. And yes, mostly they are relatively close to each other, but sometimes two double-strand breaks may be 4 microns apart, and they find each other and make a translocation. I think these are very, very interesting experiments, and because you get proof which is undoubtable. And we are not so far as we should be, and we should take serious our limitations. It's very difficult to say something about a very specific experiment without knowing all the details, really.
So the last question to Professor Silvio Onesti.
Mine is a much more general question. You talked about evolution at the beginning, and I was wondering if anyone has ever looked at archaeal nuclei to see if there is any evidence already of some clustering?
Yeah, I would like to be among the first who do that. Yeah. No, people, there is electron microscopy or so, but not systematic studies. Recently, the Meshima group published a paper in PLOS ONE telling that, I mean, but it was technically very well done, I think, that when you have clustered chromatin, you get less DNA damage. And in evolutionary terms, I think the following. In early evolution of archaea and eukaryotes, I mean, the repair systems were certainly not that perfect than today. Now, when you cluster the chromatin, it's like on a battlefield. If everybody is down, maybe it can less easily be hurt. Yeah. On the other hand, you cannot act by only waiting. You have to expand or so. The chromatin has to do something. But maybe there's a balance. Maybe there was a selective force on that kind of clustering, and the mechanisms may be different. But that's the reason for me. An archaea is an outgroup, yeah. And when you want to understand and you believe that nuclei did not fall from heaven, yeah, maybe some of the principles were already developed in the prokaryotes, and I would love to know that.
Okay, so while reminding again you that Professor Cremer is around for the rest of the afternoon, I'd like to close the session and also thank Professor Cremer for having accepted the kind invitation to come and give the colloquium. Thank you very much again.